country because knowledge belongs to the humanity and it is a torch which illuminates the world. It is my pleasure and pride to welcome and introduce our guest. That is, we are extremely honored to have a keynote speaker of a today's program, Dr. Sirina ma'am, and resource person of the today's program, Dr. Rekha Magirwar ma'am, and the chairperson and our mentor and respected principal sir of our college, Dr. Shodha Srisat sir, and vice principal of our college, Ramesh Shinde sir. May I request to everyone to give them a warm welcome. And now I would like to request our respected principal sir, Dr. Shodha Srisat sir, to give us guidance.
गुड मॉर्निंग, नमस्कार. MSP मंडल यशवंतराव चव्हाण कॉलेज अंबाजोगाई, डिस्ट्रिक्ट बीड, महाराष्ट्र. डिपार्टमेंट ऑफ माइक्रोबायोलॉजी organized one-day national online seminar on dual application of microbial technology, mycorrhizal sustainability, and Azurin-based anti-cancer potential. I am grateful to all Honorable Executive Council and CDC members. The office bearers of all the organizations have given their best wishes for this seminar. Today, our guest of honor and keynote address of this seminar, Honorable Dr. Serena, Assistant Professor, Department of Life Sciences, Calicut University, Kerala. Our other resource persons, Honorable Dr. Rekha Magirwar, Professor, Department of Botany, Sri Shivaji Science College, Amravati. Convener of this seminar, Miss Pradnya Wavadkar and Miss Poonam Lakhe, and organizing committee members. My college vice principal, Dr. Ramesh Shinde, and Mr. P.K. Jadhav. Technical supporter, Mr. Ali Sheikh and Mr. Asif Sheikh. All my teaching and non-teaching staff member and across the participants. Dear participants, today I am going to talk about a very important, exciting topic, 12 applications of microbial technology, mycorrhizal sustainability, and Azurin-based anti-cancer potential. I am a student of language and this subject is a new to me. The topic of today's seminar is very important. Today, we are connecting two seemingly unrelated fields, sustainable agriculture and oncology, from the perspective of microbial innovations. The first part of this seminar, the green pillar, mycorrhizal sustainability. Our first focus is on arbuscular mycorrhizal fungi, the silent partners of the plant kingdom. As we grapple with global soil degradations and climate instability, mycorrhizal technology offers a path to true sustainability. By forming a symbolic relationship with plant roots, these fungi act as a biological extension of the root systems. They not only enhance nutrients uptake, they also improve soil structure through the production of glomalin, increase drought resistance, and reduce our dependence on chemical fertilizers. In the age of climate smart agriculture, mycorrhizal are not just a tool, they are a necessity for food security. The second part of this seminar is the clinical pillar, Azurin-based anti-cancer potential. Shifting from the soil to the laboratory, we look at azurin, a blue copper protein produced by the bacterium Pseudomonas aeruginosa. We often view bacteria through the lens of infections, but Azurin showcases the therapeutic side of microbiology. What makes Azurin revolutionary is its selectivity. It preferentially cancer cell without harming healthy cells. Once inside, the stabilize the P53 tumor suppressor protein effectively, reacting the cell's natural ability to undergo apoptosis. The dual action playing both the role of delivery vehicle and therapeutic agents represent a shift towards non-toxic targeted cancer treatments. The common threads today is innovations through symbiosis, whether the fungi helps a plant survive drought or a bacterial protein help the human body fight tumors. Microbial technology is proving that the smallest organisms can offer solutions to our biggest problem. This is the magic of this subject. I hope that this seminar today will not only serve as a platform for exchanging informations but also for fostering interdisciplinary collaborations that will shape the next decade of biotechnology. Once again, thank you all participants.
Thank you so much, sir. It is a really proud and honor for our college to organize such a beautiful program with such an amazing guest. Words are too short to describe their achievement. Now, I request to my colleague, Ms. Kople Tejaswini ma'am, to introduce our today's keynote speaker, Honorable and respected Dr. Sirina ma'am.
Good morning, everyone. It is my great pleasure to introduce our distinguished guest for today's seminar, Dr. Serena MC Ma'am. Ma'am holds PhD in microbiology from the University of Calicut, Kerala. With her doctoral research focusing on cancer growth suppression using purified azurin from native Pseudomonas isolate, her work lies at the intersection of applied microbiology, molecular biology, and cancer research, making her a respected researcher in the field. She has an impressive academic background with master in microbiology from MES College, Aluva, and Bachelor's of St. Mary's College, Thrissur. Ma'am has published multiple research papers in reputed international journals, including Springer, Nature publications, and has contributed books, chapters, and conference proceeding at both national and international levels. Ma'am is ICAR NET qualified and has received recognition for her research presentation, including the Best Paper Presentation Award at national and international conference. She actively participated in the numerous workshops, conference, and training programs in microbiology. Academically, she has served as a assistant professor of microbiology at reputed institutions such as Royal Dental College, St. Joseph's College, and the Department of Life Science, University of Calicut. Her teaching and research experience spans microbial, microbial techniques, molecular diagnosis, cancer cell, the studies, and industrial microbiology. Today, we are fortunate to have her with us to share knowledge and insight with us. Thank you.
Thank you so much, Kople ma'am. To know more about today's topic, organized by the Department of Microbiology, I like to request Honorable and respected Shirina ma'am to deliver their speech and guide all of us.
Thank you. Good morning, all. Myself,
Good morning.
Professor. I'm currently working as assistant professor in the Department of Life Science, University of Calicut, Kerala. I am very happy to share my knowledge here and happy to take a part of this seminar. Thank you. So let's go to the session.
Ma'am. Hello.
Yes, 1 minute.
Ma'am, is any slide sharing problem? Then send PP to me. Our sir show it on PC. Hello? Hello, ma'am?
Yeah, a certain slide sharing problem. Give me 5 minutes.
Okay, ma'am. Okay. Sorry for inconvenience. Due to some reason, ma'am share her PPT slide with me. Ali sir,
Yes, madam.
display it on your PC. Okay.
Okay, please send me, madam, PPT.
Yes, yes. Sorry for inconvenience, all of you. Sir, can we start now?
Yes, madam.
Okay. Ma'am, may I audible to you?
Yes, audible.
Okay.
Okay, can you share the slides, please? I will explain. I will teach.
Yes, ma'am. 1 minute.
Okay.
मैडम स्लाइड ओपन हुई दिख रही है क्या आपको पूनम मैम
हा सर. नाही सर, स्क्रीन दिसत आहे फक्त.
ओके ओके जस्ट
Okay.
Yes, sir.
Can you please open the previous slide? That's the title slide. Okay, let's start. Actually, my research work on the. Are you audible? Or
Yes, ma'am. You are
okay,
audible. Yes.
okay. My research work is mainly focused on the anti-cancer therapies. Cancer growth suppression using purified azurin from native Pseudomonas isolates. That's my research topic. Next slide, please. Actually, as we all know, cancer is a critical health condition as it affects almost every tissue and organ of the body. So, cancer is considered as the second leading cause of death after heart diseases. So, prevention of cancer is the most important health challenge before this scenario. So, discovery of new anti-cancer agents with high efficiency is the need of this hour. Okay, next. That's the current situation of cancer. Hence, the remedy for a cancer is bacterial therapy of cancer. That means live or attenuated pathogenic bacteria or any other products extracted from the bacteria can be used as a remedy or a treatment therapy for cancer. So, researchers looking to engineer microorganisms to produce medicinally important compounds and to improve their activity via protein engineering, and cancer treatment only become more economical and available. As we all know, most of the people cannot afford the cancer treatment. So these studies may be a remedy for that such people. Okay, next slide. So mainly I am focusing on a small protein that is very, very important, azurin. It is a small cuprous protein produced by Pseudomonas aeruginosa. Its cytotoxicity is only for specific cancer cell. That is its very peculiarity. It does not affect the healthy cells. The neighboring healthy cells will not affect it. A number of therapeutic substances can link to azurin, so it can act as a cargo protein. Okay, next. So Azurin also have some abilities that is anti-malarial, anti-HIV, anti-parasitic, and anti-cancer. These are the major properties of Azurin have. So next slide. It is a multi-phased anti-cancer agent. As we all know, cancer is a complex procedure. So Azurin acts in mainly 3 ways to fight against the cancer. First one, it induce apoptosis through intracellular stabilization of P53 protein. As we all know, P53 is a tumor suppressor protein. Second type of that is it inhibits the cell cycle progression through intracellular binding to F receptors tyrosine kinase. Third one, it prevents the angiogenesis through inhibition of vascular endothelium growth factor A. Angiogenesis means it is a process to produce more blood vessels to the particular area which is transformed into cancer. So, azurin prevents the angiogenesis. These are the 3 mechanisms proposed by the azurin to fight against the cancer. Okay, next one. Next research problem. Next one. Next slide. Major objectives of my slide is first one, isolation and identification of azurin gene harboring Pseudomonas isolates from Malabar region. It's from Kerala. It's the middle part of Kerala. Second, my objective is to cloning, expression, and purification. The particular azurin gene is extracted and is done cloning. Lastly, the study of growth suppression by azurin on various cancer cells. Okay, next one. Firstly, the first objective is isolation. Next slide. Isolation. Actually, isolation and identification of Pseudomonas isolates from environmental samples of Malabar region, such as water, soil, etc. Then cytotoxicity of isolated Pseudomonas strains were measured by trypan blue exclusion assay. It is a assay used to detect if the cell is viable or not. Then it is detected by the Azurin gene detected by PCR, which is very sensitive technique. As we all know, polymerase chain reaction, which is mainly used to detect the presence of azurin by the particular primers. Okay, next one. My methodologies are isolation of samples and identification. Isolation by from soil and water, identification by, as we all know, the PCR. Then extracellular protein extraction. That means by protein extraction by ammonium sulfate precipitation, dialysis, and cellulose treatment. Then trypan blue exclusion assay. That means the ability of the bacterial substance to kill some cells. That is the trypan blue exclusion assay. Then PCR reaction. That is the methodology primarily applied for the isolation of azurin containing bacterial strains from environmental samples. Okay, next one. And the protein profile done by SGS page and comparison study with a standard strain. Standard strain means it is already MTCC strain which is studied by the Azurin-Based. So my samples were compared with the MTCC strain and its characterization of the partially purified Azurin was done by FTIR Malitov. And MCF cell lines were used to study the Huston staining method. So these are the primary set of works used to identify the ability of azurin from environmental samples. As we all know, the extraction of azurin from environmental samples are very scarce in the scenario of India. Okay, next. Next about the research. Actually, I isolate 43 isolates in which cytotoxicity can be done by 10 partially purified bacterial protein extracts was carried out. Actually, trypan blue exclusion assay which produce cytotoxicity. That means the 10 strains have the capacity to kill other cells. That means cytotoxicity. But strain A, which shows highest activity, that is 85% when compared to other strains. Next slide. This is another graphical representation of trypan blue exclusion assay for cytotoxicity. That means the first line, blue line, which shows 85% cytotoxicity. That is the sample A. Next one. This is the PCR. Out of these 10 samples, when we done PCR with the Azurin primers, forward reverse primer, the length is 545 base pair. It is present shows in 8 isolates. Actually, C and B, which didn't show any Azurin as we conducted PCR reaction. Under 8 strains which produces a clear band, that means they contain azurin gene. Also contain cytotoxicity activity. Okay, next. This is the SGS page for the profiling of protein. The total partially purified protein extracted from 8 azurin produced isolates. The 8 isolates were chosen by PCR reaction and it produced almost 14 kilodalton. That is the molecular weight of the azurin, which produce a clear band at the same site of 14 kilodalton molecular weight was present in all 8 azurin gene harboring isolates. Next one. This is the molecular characterization and the strain with the highest cytotoxicity was further chosen for the study, which is assigned the Pseudomonas aeruginosa SS strain, which forms a characteristic family tree or molecular analysis tree. Okay, it is also deposited on the GenBank to give a accession number. Okay, next one. So, 16S rRNA gene sequence was deposited in GenBank database with the accession number KU821118. That is the novel strain which I isolated from the soil, which harbors azurin gene with the highest cytotoxic activity. Okay, next one. Azurin gene sequencing also deposed on GenBank with the accession number KU82119. Azurin gene sequence I have been done by PCR. It's also deposed on GenBank. It's very novel technique. When we isolate some bacterial strains with a novel characterization, we have to sequence their gene, and we can deposit on GenBank, so we can get a accession number for that particular strain. Okay, next one. Comparison with the Pseudomonas aeruginosa MTCC 2453. Actually, MTCC 2453, which is already studied, Azurin containing strain, which is already studied. So I next did comparison with the already studied strain with my isolated strain, which also shows some similarities, almost same. Okay, next one. FGR characterization was done, Fourier transformed infrared characterization, which give a detailed information about the chemical structures present in the protein. It shows a deep grooving at 1063H, means it contains C double bond O bond. That means it contains azurin. This is a very particular study in the base of chemistry, FGR studies. Next one. So FGR analysis of protein samples from the standard strain also show the same peak around the same sites, that is 1637 centimeter. My strains which has got 1638 centimeters. So it was almost similar when we compared with the standard strain. Okay, next one. This is the standard strain FGR analysis. Next one. Malditoff analysis. Malditoff analysis done by which contains a mixture of proteins. It's almost same as the STS page technique. That means it detects the presence of protein. It is a partially purified protein. So it also contains some another protein, but it contains a 14 kilodalton protein. That means for this partially purified extract contains some azurin. Next one. Hashard staining. Molecular characteristics of MCF breast cancer cells after partially purified protein sample observed by Hashard staining. The protein treated cell showed nuclear condensation. That means cells are killed by the action of this particular protein suspension. That means it has the activity. That is the Hashard staining method. This is the preliminary studies of azurin containing strain, which I deposed on the GenBank, and I named it Pseudomonas aeruginosa SSJ. So next, my objective is to how can we overproduced or highly produced azurin from this particular samples, how we clone that particular strain and produce a lot of azurin with a high activity. Okay, next slide. So the preliminary study provided evidence for the existence of azurin harboring Pseudomonas strains in Kerala. A novel strain of Pseudomonas aeruginosa SSJ, which produced promising levels of active azurin, was successfully isolated, and characterization study was also done in the primary stage. Next. These are the papers published depending on this data. Next one. Okay, next. Next, my objective is to cloning, expression, purification, and characterization of azurin. That means the azurin genes are also present. So we have to clone that particular gene. So azurin is abundantly produced. Next one, next slide. Next. Cloning of azu gene in PET22B vector and expressed in E. coli BL21. Characterization of purified azurin was using again FGR, Malditoff, and LC-MSMS. Comparison studies of purified azurin with the standard azurin, which we had purchased from Sigma Aldrich. Actually, as we all know, azurin is very costly. For 1 milligram, it costs about 80,000 for 1 milligram. Docking studies of the azurin with some anti-cancer ligands also done. These are the objectives in the second stage. Next. Cloning of azu gene is done in E. coli DH5 alpha, expression host E. coli BL21, and PET22B is the vector. Restriction enzymes hence we used HIND3 and BAM H1. So this is the cloning procedure. This is very complicated procedure. After many trial and errors, we expressed the Azurin gene. Next. Firstly, DNA isolated and primer designed by the software primer 3 and PCR was done and purified the PCR product. Restriction digestion done by restriction enzymes and alkaline phosphatase treatment. Competent cells are prepared and transformed and colony PCR was done. Okay, next. Actually, the express protein extracted and STS page was done and purification was done by nickel NTA column. This is a very peculiar technology to purify recombinant proteins only. Otherwise, you have to use some cephaldogs or column chromatography. In the case of cloning and recombination, we have to use this nickel NTA column. Then characterization of azurin by FGR, Malditov, LC-MS. And some docking studies are also performed by bioinformatics tools. Next one. So genomic DNA was extracted. PCR amplification of Azurin genome Pseudomonas was done using specific primers. PET22B vector and purified PCR products were digested using EcoR1 hint 3. These are restriction enzymes. And they are transformed into E. coli DH5 alpha strains. Next. Few randomly selected colonies were selected for colony PCR. The plasmids from the transformed colonies were isolated and digested. On double digestion, the recombinant plasmid two bands correspond to the vector and insect were observed. We have to identify the insert is rightly inserted into the vector. Next. Then it is amplified and expressed. The amplicon of 1224 base pair was sequenced and flagging regions were removed. The cloned sequence were also sequenced and deposited in GenBank with the accession number 12411. That means already in the first stage, the isolated a strain from environmental sample. It is sequenced and it is also deposited in GenBank. After cloning from the Azu gene from the environmental sample is cloned and recombinant gene also sequenced and deposited in GenBank with a different accession number. Okay. Next. STS page is also performed. It gives a clear band in the 14 kilodalton weight. So STS page is mainly used to done for identification of protein with respect to molecular weight. Okay. Next. FGR also performed with the standard Azurin. So it almost shows similar peaks around 1600 range. That means it also contains C double bond or bond. Same chemical bonds are present. Okay. These are all characterization studies. Next. Malditov. In a peak was observed around 14 kilodalton molecular weights, which assumed the presence of azurin. Malditov is very sensitive technique. Next one. LC-MS. MS spectra analysis of azurin showed 4 short peptide fragments. The protein sequence of these short peptides were compared with the azurin sequence obtained in this study. The sequences were performed and it shows 100% identical to the standard azurin sequence. In the technique of LC-MS, we sequenced our recombinant or cloned and purified products. It shows 100% similarity with the standard azurin. Next. These are the fragments. Next one. These are molecular docking results. Protein sequence shows 100% similarity with PDB-AD1 VLX. It is docking studies and all receptors gave good docking score. That means this gene sequence hit with a cancer ligands in the sense of bioinformatics software, which shows highest activity in between these two. That means they have high anti-cancer activity. Okay. In dry lab. Next. These are the interaction between receptors and azurin. Visualization of dog complex by PyMol software. Next one. So in the second chapter, we report the successful cloning and expression of azurin gene from Pseudomonas aeruginosa SSJ. And characterization studies of recombinant azurin was also done by FGR, Malitov, LC-MSMS. And docking studies of Azurin were also done with selected breast cancer cell receptors by bioinformatics tool. So it would be used for anti-cancer studies. So it is highly effective. Next to the last unit, next one. These are the papers published based on this data. Next. Study of growth suppression by Azurin on various cancer cell lines. As we all know, we firstly isolated an environmental sample. It shows highest cytotoxic activity. So we cloned that particular gene and produce highly active Azurins. Next, we applied application level, that is the third level. We studied its efficiency on various cancer cell lines. And also we detected its efficiency on normal cell lines. That means it does not harmful to the normal cell, but it efficiently killed the cancer cell. Okay, these are the next topics. Next. Next. So detection of the cytotoxicity of recombinant Azurin and stranded Azurin on different cell lines. Here we used mainly 3 types of cells. First one, human breast cancer cell, MCF7, then human colon carcinoma cell, HCG15, and normal cell lines, Biro cell lines. Determination of apoptotic efficiency of Azurin by Hash's staining by FRET-based assay. Then also, after using cancer cell lines, we performed in vivo anti-tumor assay. So on mice. Okay, next. 5-pan blue exclusion assay was performed and MTT assay was also performed on 3 cell lines. MTT assay is more confirmed test when compared to trypan blue. The cells were incubated for 48 hours in a carbon dioxide incubator in the case of MTT assay. Next. Hash's training, here we used colon carcinoma cells, which is very complicated work to detect the cancer growth suppression is focused in which stage of cell cycle. We have to identify in which stage the Azurin is worked properly. Next. Next slide, please. Okay, the study was approved by the Institutional Animal Ethical Committee. It is the in vivo ascites anti-tumor cell. We have used experimental animals. We chosen 3 groups. First one, control group. Second group, we gave the common anti-cancer drugs. In the third group, we gave Azurin. It is a 10 days intraperitoneal injection. Next. Then the next technique is hemolytic assay. The Azurin which cause any hemolysis of the patients. So we have used absorbance of the mixture was measured at 450 nanometer after incubation. Next. Results. The MTT assay and trypan blue assay which produce highest cytotoxicity. That means the recombinant purified Azurin which shows highest activity against cancer cells. It has enough ability to kill cancer cells. Next. These are the results of MTT assay on various cell lines. First one, HCT cell line. Second one, MCF. Next one, Biro cell size. Biro cell lines, which shows cytotoxicity is reduced. That means it is a normal cell line. Next one, apoptosis assay. Apoptosis assay induced by Azurin on SW480 cells by Hash's staining method. Azurin treated cells show decreased DNA content in G1 phase and increased in G0 phase, which indicates Azurin induces apoptosis in these cells. That means DNA content is decreased. It is due to the effect of Azurin on that particular cancer cells. Next one. FRET-based assay. It is a very Casper's means assay. Separation of both fluoropore leads to the FRET loss between probes. When Azurin concentration increases, the number of apoptotic cells were increased. When we injected or we provide Azurin to cells, the fluorescence is also increased according to the killing of cancer cells. If killing does not occur, the risk of fluorescence occurs. This is the basic principle of this technique. We have to identify the concentrative efficiency of the particular anti-cancer drug by this FRET assay. Okay, next one. Apoptotic potential of azurin was confirmed in colon cancer cells, which is the probe-based assay. The image gives the actual idea about the cancer cells killing ratio. Next one. French loss occurs due to a caspase activation. The staining pattern indicated the cause of cell death in colon cancer cell was apoptosis. Apoptosis means cell death. Cell death is induced by mainly azurin. The percentage of apoptotic cells are also calculated and graphically represented. Okay, next one. This is the in vivo ascites anti-tumor assay. We employed for studying the increase in lifespan of tumor-bearing animals for 50 days. Revealed that azurin-treated animals showed increased lifespan when compared to another group. Here we also made this study by grouping 3 groups. First one, control group. It does not have any treatment. Second group is took standardized anti-cancer drug, that is cyclophosphamide. The third group, that is azurin treated group. When compared these 3 groups, the azurin treated animals shows highest lifespan. Okay, next. Hemolytic activity assay. Hemolytic activity of purified azurin protein was screened against normal human erythrocytes. It showed negligible hemolysis when compared to AAPH, that is the hemolytic drug. When we treat the cells with or body with azurin, it produces normal hemolytic activity. Okay, next one. Conclusion that we report the anti-cancer activity of azurin from Pseudomonas aeruginosa SSJ expressed on PET22B vector. Cell cycle analysis, apoptosis, and anti-hemolytic assays were used to confirm azurin's potential to be used as a good therapeutic agent in cancer therapy. Okay, next. These are the international papers published based on this data. Next. So finally, in my research, we successfully isolating azurin producing strains from soil and water samples of Malabar region, Kerala, and partially purified azurin from strains SSJ was characterized and compared with a standard strain, that is Pseudomonas aeruginosa MTCC, which was we purchased. Azurin gene was cloned and overexpressed in E. coli BL21, and anti-cancer activity of purified azurins done on different cancer cell lines. And it's also compared with the standard azurin that we purchased from Sigma Aldrich. Okay, next. Then apoptotic effect of recombinant azurin was assayed by Hush's staining and FRET-based assay, and hemolytic activity also we detected. And azurin from Pseudomonas aeruginosa expressed has anti-tumor potentialities and should be further exposed in cancer therapies. Next. So these are the publications. Thank you. Have you had any queries about this? Please ask.
If any question from anyone, ask to the ma'am. Thank you so much, ma'am, for sharing your expertise experience with us. As your guidance has been valuable on your research work, it will be very beneficial for all our students and we also. Thank you so much once again.
Thank you. I'm very happy to be a part of this seminar. Thank you.
Now, again, I request to Kopile ma'am to introduce our next resource person, Dr. Rekha ma'am. Kopile ma'am, please.
Good afternoon, everyone. It is great pleasure to introduce our esteemed guest for today's seminar, Professor Rekha Magirwar, ma'am. Ma'am is the professor of botany and head of the department. She has over 19 years of teaching experience and 25 years of research experience with expertise in cytology, genetics, plant biotechnology, and mycorrhizal biotechnology. She is recognized as PhD research guide at Sant Gadge Baba Amravati University and has made significant academic contributions through more than 40 research publications, numerous conference presentation and book chapters. She has also successfully completed major and minor UGC funded research projects. Professor Magirwar ma'am has delivered over 30 invited talks, served in various academic and administrative roles, and actively contributed to student development, environmental initiatives and extension activities. We are honored to have her with us today to share her valuable insights. Please join me in warmly welcoming Professor Rekha Magriwat ma'am. Thank you.
Thank you so much, Kopile ma'am. Now I will request to Honorable Resource Person of today's program, Professor Dr. Rekha ma'am, to share the knowledge with us about their topic, Mycorrhizal Biotechnology for Sustainable Development. Rekha ma'am, please.
Yeah. Am I audible?
Yes, ma'am.
Yeah. Thank you so much, Poonam madam. At the outset, let me congratulate the initiative of Yashwantrao Chavan College, Ambezogai, for this one-day national seminar, online one-day national seminar. Dignitaries present for this online meeting. Honorable Principal of the College, Dr. Shivdas Shrisat. The keynote speaker, Dr. Sirina Madam. The organizer or convener, Mrs. Pradnya Vavalkar, Dr. Poonam Madam, and all the faculty members, dear students who have joined through this meeting. And I must thank to the director of MITSC and the president of Karyarkatta Maharashtra, Honorable Yashwanta Shitole, for introducing me to this platform. So really it's a pleasure to be the part of this national online seminar. Please allow me to share my screen.
Okay, ma'am.
Yeah, is my screen visible?
Yes, ma'am. Visible.
Yeah, is it full screen right now?
Yes, ma'am. Full screen.
Yeah, thank you. So this is Dr. Rekha Girwar, working in the Department of Botany. And you can see in this slide the credentials of our college. So it is really proud to mention that our college is the first 100 for the NIRF ranking in Maharashtra, recognized as a plus college accredited by NAC. And right now we are running two vital projects in our college, the DBT Star and PM Usha. So it's a pleasure to work in this environment and to share my insight, particularly on the mycorrhizal biotechnology and its role in sustainable development with special reference to the soil health and the agriculture and ecosystem restoration. Actually, the human life would not have existed without these essential services provided by nature. So actually, these include the clean air, and I could underline the word clean air because we are living in Amravati and it is the cleanest city in India. The fertile soil, the edible plants, and the natural regenerative processes. However, with increasing anthropogenic pressure, I can say, these natural services are under the serious threat. So you must be knowing that Sadhguru before few days he has initiated one project. He has visited many countries, and the motto was to save our soil. So here you can see the ecosystem. So these are actually the habitat, and these habitat actually are maintained, self-regulated, one can say, where the biodiversity is maintained through the natural process or the principle of the survival of the fittest. And the mature ecosystem efficiently recycles the nutrients between biotic and abiotic components, ensuring the long-term sustainability. And I would like to focus on this slide. Since schooling, we are aware of this food web, and very much acquainted with this chain. So, the discovery of agriculture marked the beginning of the civilization. However, the modern agriculture has disturbed the ecological balance, I could say, due to the excessive dependence on the chemical inputs. And we have experienced, or rather the India has witnessed, the continuous rise in the use of the different fertilizers. And at that time, the green revolution, where one can say जहाँ डाल डाल पे सोने की चिड़िया करती है बसेरा, वो भारत देश है मेरा. We have experienced this green revolution, and there was the food security for all of us. No doubt this was the golden period in our country. But right now, there is a little bit different picture, and it is a alarming picture for all of us. And where these are the alarming rates, where these inputs of the fertilizers and the pesticides, they have actually increased the productivity. Their long term use has led to the soil degradation, nutrient imbalance, and declining the main focus of my research, which I am involved since last 2 decades, that is declining the microbial activity. And it is clearly reflected in this state-wise, you can see the fertilizer consumption data. So you can see the total picture of the India, where these days we are so much dependent on the usage of the pesticide and fertilizers to get the crop in a, or the yield, which is the higher yield in a short duration. But the other side of coin is that we are losing the health of our soils and degrading these soils day by day. So this is, excuse me, soil salinity is actually the major agricultural constraint, particularly in arid and semi-arid regions. Excess salt, you can see in this picture, this is the excess salt which has accumulated in actually the root zone also. It affects the plant metabolism, it reduces the growth and may ultimately lead to the crop failure. And in India alone, nearly 12 million hectares of agricultural land is salt affected. Here you can see the overuse of the chemical fertilizers. Actually, that also leads to the reduced humus content and microbial diversity. Excess nitrogen increases the susceptibility to the pest, while phosphorus interferes with micronutrient uptake, and hence the sustainable alternatives are urgently required. So this is how there is a need of the another parallel, you can see the system which can be the eco-friendly system to overcome these problems. So such a salinity is found in our region also. You can see this is the map nearby Amravati, which is located in the region which is Purna River sub-basin. So this is known as the saline tract of Amravati region. And likewise, the health card of human being these days are we need to prepare the health card of the soil itself, and there is urgent need, as I have just mentioned, to have the system which could be considered as a eco-friendly environment for the sustainable development and to get the crops in a better manner. Or really, it's a pride for me to mention that I am working in this area, which is actually beneficial and which made the entire country and the world also, I could say, that these are the tools of the biotechnology. The mycorrhizal fungi, they are the boon for the you can say the different crops and the plant world. So these are some of the news published in the daily, and yes, I'm working in this area since last 2 decades. Right now 5 students are working under my guidance, and many PG students have submitted their projects under this study of the arbuscular mycorrhizal fungi. So here you can see where the regarding the distribution, these are ubiquitous, and the main thing is that they are the major symbiotic association with the plant world, and it is said that it is difficult to find the plant which is not having this symbiotic association. So apart from the arbuscular mycorrhizal fungi, these are the different mycorrhiza, and they're specific with the plants belonging to the certain families. And yeah, I must mention here that this slide is particularly focusing on the characteristic features that these fungi have, and these structures actually confirm the presence or the association of these mycorrhizal fungi with the host plant. So there is a such hyphal network, and it is considered as the extended arm of for the roots. So it travels to the certain area in the soil itself, and this is the extended arm for the absorption of the water and the mineral nutrients. I think someone is disturbing the screen. Organizers, please take care. These are the viable spores, mycorrhizal spores. So these are actually the tools which can be multiplied and which are the source of the mycorrhizal association. There are vesicles and the arbuscules which are seen in this slide. So this is the hyphal network, and these are the minute thread-like structures traveling up to certain area and working as an extended arm. So there are actually two types, Paris type of the association, which are inserted inside the cell, and this is actually the cortical region of the host plant root, and arum type of the arbuscules you can see here. So these are the pictures taken by our students in our research lab. So there, these slides are explaining the mechanism of the association of this mycorrhizal fungi. So this swollen structure is actually the appresorium through which the mycorrhizal spore germinates, and after germination, the hyphal entry occurs inside the host root. So this is the pictorial view where you can see how the hyphal network is extended outside the host root as well as inside the cortical region. And here you can see the network of the hypha, and there is arbuscules which are developed after some time, and arbuscules are actually the fungus and plant metabolite exchange site. So where the plant provides the ready-made food material to the fungi, and fungi in return gives the important phosphate as well as the micronutrient copper and the zinc. So these are the small bush-like structures, the arbuscules. These are the vesicles which are having the hyphal connections. These are the beautiful spores isolated from the soil. So there is right now the renowned mycorrhizologist from India. We rely on the morphotaxonomic identification, but one of my research paper was rejected on the note by the Journal of the United Kingdom that you must adopt the molecular identification techniques, and these are quite expensive, and one must need the sophisticated laboratories for this molecular identification. So in future, my students may go for this molecular identification technique. So these are the criteria which are actually considered for the identification of different genera of the mycorrhizal fungi. We consider the size, and recently there are lot of advanced microscopes which are directly connected to the laptops or the computers, and one can have the image of these isolated spores, and we can measure the size, we could know the shape, color, and the wall layers are considered quite important, the content and the subtending hypha and the pore. So these are actually different criteria for the taxonomy, morphotaxonomic identification. So these are the pictures which our students have taken. So in our region so far in last 20 years we have encountered majority of the glomus species. So this is glomus fasciculatum and sclerosistes. So these are the important genera of the mycorrhizal fungi: Acolospora, Enterophospora, Digaspora, which is showing here this bulbous suspensor, and the Scutellospora. You can see here the germinating shield is the one of the identification criteria. Yeah, this one of the important aspect that I have encountered, and I have presented this paper in Asian Association, the conference of mycology, which was held in South Korea, Incheon, South Korea. I have presented this paper. This is actually known as the spore syndrome. So this is actually one dead spore can serve as a micro habitat for the other spores. So as you can see in this picture, this is the outer spore, and inside you can see the small other spores which are the living ones. And so far the research has proved, and the literature also has mentioned, that there are a lot of vital role played by these mycorrhizal fungi in relation to the uptake and the accumulation of the phosphate and copper and zinc. They also enhance the nitrogen fixation. And one of the interesting feature that I have encountered, when the plants belonging to the family Fabaceae, they are associated with other family plants, then there is an intermingling of the roots and the root nodules, which is a characteristic feature of the Fabaceae or Leguminosae plants. So that was also observed on the roots of the other plants, which is closely associated with the such plants. So one of my research students have encountered such situation, and these roots actually are traverse to up to certain distance, and they intermingle the roots of different plants, and they also increase the nutrients and water absorption area. So they also provide the growth hormones and increase the tolerance of the plant. So these are the biological controlling agent. So they, once the mycorrhiza is associated with the host plant, the other pathogens like nematode can't infect the plant. So as you can see in this cartoon, when the roots underground part of the plant is healthy and happily growing, of course the output is that the above ground part is also growing in a healthy manner. And this is the phosphate uptake. You can see this is the yeah, you can see here how how the molecular mechanism takes place, and the phosphate from the soil, how it travels. Actually, the enzyme which is necessary to release the phosphate resides inside the mycorrhizal fungi only, and it releases the phosphate to the host plant cell. Yeah, these are some of the other roles played by these fungi. So when we actually plan the work in mycorrhizal biotechnology, this is the plan that we follow. Firstly, we have selected the degraded saline soils from our region. We have done the parameters to know the physicochemical analysis of these soil samples, and there was quantitative as well as qualitative estimation of arbuscular mycorrhizal fungi. And we have isolated, as you can see, the ubiquitous fungi, which we have isolated through wet sieving and decanting technique. And then we have taken the pictures and we have done its morphotaxonomical identification. Then we have worked on the spore population, and we have find out which is the dominant mycorrhizal fungi in that particular area. Then we have come across with its to maintain its pure inoculum for the mass multiplication. And I have worked on many crop plants to know the actually the effect of arbuscular mycorrhizal fungi, and we have compared it with the chemical fertilizers, a single superphosphate or other chemical fertilizers, and there are promising results since last 2 decades that these mycorrhizal fungi, actually the native dominant mycorrhizal fungi, can be the effective biofertile, can be used, can be applied as a effective biofertilizers for the crop production, and reduces the cost which our actually farmers could not afford the cost of the chemical fertilizers. And that is why this is the bio tool, one can say. So this is the methodology. You can say this is the wet sieving and decanting technique, and on this petri plate you can see the isolated spores. So here you can see through this slide, these are the beautiful mycorrhizal spores which are isolated from the soil. And same time, we need to confirm the association in the root. So this is the method where we can see the roots are treated to find out structures like vesicle, arbuscules, and spores, whether to confirm the association of the mycorrhizal spores with the root, host plant root. So far our research is actually has confirmed that there are the different plants which are associated with the mycorrhizal fungi. We have raised the mycorrhizal pure as well as mass multiplied dominant mycorrhizal fungi, and the growth parameter on soybean, that is glycine max, Vigna radiata. So these are the techniques which are the eco-friendly techniques and which can certainly be applied to reclamation, the strategies for the saline degraded soil from our region. And so what is actually required for the management of these mycorrhizal fungi? That most of the time we need to protect the topsoil and the microbial activity. And as I have mentioned in my earlier slides, that the anthropogenic activities and the excess utilization of the chemical fertilizers, pesticides, are actually the threat for these microbial activity in the topsoil region. So do not use the chemicals that actually killing these mycorrhizal fungi. So do not also over fertilize the soils, and so that it is having the biomagnification effect also. So maintain the keep the native plants in your region. So these are some of the photographs where we have compared the control plants with the treated, that is the non-mycorrhizal and the mycorrhizal plant. So this is the funnel technique, wherein we have mass multiplied the mycorrhizal, because one thing I would like to mention, that very meager research is carried out and it is not that much successful to mass multiply these propagules on the exenic culture on different media like MS medium or the different media under tissue culture. So these mycorrhizal spores, they mass multiply in soil itself. So we need to propagate them, mass multiply them in the soil medium. So these are the funnel technique or the pure inoculum. And I have one of the student have worked on the, we have tried the strawberries in our region, the Melghat tiger region near to our city, and we have studied the mycorrhiza associated with these strawberry plants also. This is the soil trap culture we are maintaining our lab. Actually, the energy Terry has developed and brought these products in the market. So this can be, dear students, this can be the idea for startup in your region, as the native and dominant mycorrhizal fungi from your region can be served, can be applied as a biofertilizer. So you can maintain the pure cultures and mass multiply them, and in soil medium you can bring it to the market and you can supply the it to the farmers. You can train the farmers also, and they can raise their biofertilizer in their fields themselves, and they can reduce the cost of the chemical fertilizers. So these two products are brought by the energy and resource utilization. One of them, it is in the powder form and the other is in the tablet form. So that is applied in the field. So let me conclude by saying that there are a lot of mycorrhizal fungi in our Vidarbha region, and no doubt they are the phosphate fertilizer saving boon for the natural boon for the farmers, for the ecosystem, that is man-made ecosystem. Then they can be used for degrading the saline soil for the reclamation program. What so far I have encountered, that it is the Glomus fasciculatum, Glomus aggregatum. They have shown wonderful results on the crop plants, and we have tried to apply the single as well as the dual inoculum for the crop plants. And my future plan, as I have mentioned, one of the research gap in India, that we need some molecular approach for the molecular identification of these fungi, and also we need some media apart from the soil medium to mass multiply these fungi and artificial cultures of these mycorrhizal spores. And that's why the further research is required. Applied aspects are actually needed, and this Gen Z can work now on these future aspects. So let me thank the organizers for giving me this opportunity. Dear students, if you have any queries, I'll be happy to answer. Thank you. Thank you so much.
Thank you so much, ma'am. Very big thank you to ma'am, because mycorrhizal biotechnology is indeed a cornerstone of sustainable development, offering eco-friendly solution to modern agriculture and environmental challenges. Thank you so much, ma'am.
My pleasure.
Now, expressing gratitude, we must never forget that the highest appreciation is not utter words, but to live by them. So I would like to call Acharya to say vote of thanks.
Thank you, ma'am. Good afternoon, everyone. I am present here to offer my thanks of behalf of College and Microbiology Department. We would especially like to thanks the Dr. Sarina ma'am keynote speaker of today's program, and resource person, Professor Dr. Rekha ma'am, for accepting our invitation and joining us today. Really, ma'am, your presence has boosted up our energy, and your thoughts have inspired us and our students, and your valuable words motivated us. Then I appreciate thankful to our respected principal Dr. Shivdas Sesar sir for always encouraging and directing us. Thanks to Vice Principal Ramesh Shinde sir. I also thanks to all my staff members, teachers, and special thanks to technician staff member Asif sir, Ali sir. Also thanks to all audience for joining the program. All peoples who are attending this program deserves the many thanks. Thank you all again. Thank you.
Thank you so much, sir. This meeting will end here.